Hey everyone, welcome back to today's class. Now in today's class we are going to study about the transverse binary fission. We have already seen longitudinal binary fission. Now today we are going to see transverse binary fission. Where will you see transverse binary fission? You will see in Paramecium. Where we have seen longitudinal binary fission in Euglena, right? Now it is commonly called slipper animalcule. Animalcule, when I say, that means it is microscopic little organisms, right? It is a slipper-like structure, so we call it as slipper. Now, axis of cytokinesis. At which axis cytoplasm divide? Now we know that longitudinally पे we said कि axis of cytokinesis is longitudinally. But what about the axis of cytokinesis in transverse? Obviously it is going to be transverse. But आपके textbook में क्या लिखा है? It is perpendicular to the longitudinal axis of body. So this is your organism, and transverse, this way, this we call it as transverse. Isn't the transverse is perpendicular to the longitudinal one? That's what is written. Okay, so perpendicular to the longitudinal axis of the body. Transverse binary fission is also known as homothetogenic fission or perkinetal fission. When we talk about homothetogenic fission is nothing but when the plane of fission, so this is your organism and this is your plane of fission. When the plane of fission is perpendicular to the longitudinal axis, it is called as homothetogenic. But when the plane of fission is perpendicular to the kinety, what is kinety? We have seen three components will form kinety. What are the three components? Write it in the comment section and let me know. So this is your longitudinal kinety present over here, and your transverse axis will be here. So it is again perpendicular. So if the plane of fission is perpendicular to the longitudinal, we call it as homothetogenic fission. If the plane of fission is perpendicular or right angle to the kinety, we call it as perkinetal fission. Now this is the structure of Paramecium, super important, same as the structure of Euglena was super important, right? When you see the structure of Paramecium, here is the oral groove present over here, that is an opening, and all over the body you can see cilia, and outer layer we call it as pellicle, right? So cilia and pellicle, you know it already. Oral groove is nothing but groove matlab ghatta hota hai, right? So your food is going to enter through oral groove. Then we have cytostome, which is a mouth-like structure. Below that we have a muscular tube-like structure, which we call it as cytopharynx, and then these cytopharynx will lead into the food vacuole. So food is going to enter into the food vacuole, and once this food vacuole is filled, what will happen? It will pinched off from there, right? Now then we have contractile vacuole. We have only two contractile vacuole present over there, but in longitudinal we have studied only one contractile vacuole, right? So contractile vacuole which is present at the posterior side, we call it as posterior contractile vacuole. Which is present at the anterior side, we call it as anterior contractile vacuole. Then the last one we have cytophage. Cytophage is nothing but it is used for digestion. It is used for digestion. Now the super important one is your micronucleus and macronucleus. In Euglena we studied only one nucleus is there, but when we talk about the Paramecium, we have two nucleus, that is micronucleus and macronucleus. Micronucleus basically it will help in reproduction, and macronucleus it will help in metabolic activity. Which activity? Metabolic activity. Let's see, this structure is super important, class. I want all of you to make it, right? Now let's see. Pellically we already know. Contractile vacuole help in osmoregulation. What is osmoregulation? Write it in the comment box. Micronucleus is your haploid or diploid. It is diploid and controls the reproductive activity. But macronucleus is polyploidy. That means more than diploid. 2n ke alawa 3n, 4n, 5n. These all are called as polyploidy. It is going to control the metabolic activity of the cell. What protein has to be produced and all these activities, energy production, all this will taken place by macronucleus. Cytostome, you know, mouth-like structure. It helps in ingestion of the food. Cytopharynx, engulfing of the food. Food vacuole, which helps in storage and digestion. Cytophage, it helps in egestion. Then cilia, you know, ectoplasmic component. What is the meaning of ectoplasmic? We have already studied endoplasmic and ectoplasmic in infraciliary system. Ectoplasmic means outer cytoplasmic, right? Outer cytoplasmic layer, we call it as ectoplasmic. So infraciliary system we have already discussed. This one is the new one, trichocyst, which is present on the outer layer of the cytoplasm, and it helps in defense mechanism. It will save the paramecium from the foreign particles, right? Now, before moving ahead with the mechanism of transverse binary fission, we need to understand about the amitosis and mitosis. Mitosis, you already know, it is nothing but the equational division. It is nothing but the equational division. When I say equational division, so suppose if your parents are diploid, then the daughter cell will also be diploid. If the parent is haploid, the daughter cell will also be haploid, right? So equal amount of chromosome is passed, or the same amount of chromosome is passed what is present in the parent. This kind of division we call it as mitosis, and this will occur through a cycle, cell cycle. We call it as G1, S, G2 and M phase, right? It occurs through this cell cycle. So it is a long process which occurs through the cell cycle, and in the cell cycle, when we talk about the mitosis, karyokinesis followed by cytokinesis. This is what we studied in the longitudinal binary fission, right? Now, when we talk about amitosis, there is no proper cell cycle we are supposed to follow. It is nothing but, मान लो ये एक चीज है, यहाँ पे आपने लिया, इसको खींच दिया, इस तरीके से खींचो, इस तरीके से खींचो. So what will happen? There will be cleavage will be formed, and finally what will happen? It will leads to the breakage of the parent cell and leads to the formation of two daughter cell. तो इसमें क्या हुआ? मान लो कुछ अगर DNA यहाँ पे present हुआ तो इसमें आ गया, कुछ यहाँ पे present नहीं हुआ तो यहाँ नहीं आया. तो due to that what will happen? Unequal amount of genes will be transferred. So parental DNA distributed randomly. Why? Because direct cell division occur here through simple cleavage, right? These daughter cells are not genetically similar. Correct? Now let's see the mechanism of transverse binary fission. This diagram was drawn by Yagna. Beautifully, you can see here the mechanism of paramecium. Let's see here and then I'll tell you step by step. So this is the paramecium over here, and we have seen that the karyokinesis is going to be followed by cytokinesis. So first we will see the division of nucleus. How many nucleus we have? Two nucleus, macronucleus and micronucleus. When we talk about the micronucleus, micronucleus will divide through mitosis. Okay, proper cell cycle is going to be followed. So equal amount of DNA is transferred. But when we talk about macro, it is going to be divided through amitosis. That means just through the cleavage cell is going to divide. Not equal amount of genetic material is transferred to the daughter cell, right? Once this division is done, then what will happen? Here you can see the structure of spindle fiber that will help in mitotic and meiosis division. But right now you don't have to study that. Okay, it is just mentioned, so I am telling you. Now, while this is happening, what will happen? There will be the formation of new oral groove. There will be the formation of new oral groove. Now, when there is after the division of a nucleus, what will happen? We have to follow the cytoplasm. We have to follow the cytoplasmic division. We have to follow the cytoplasmic division in which way? In a transverse way. Now, this way it is going to be. So, transverse way में है तो what will happen? There will be constriction. एक दबा जैसे पिल्लो लो और पिल्लो को बीच से दबाओ तो how constriction will occur? Same way. ठीक है. अब यहाँ पे क्या होगा कि ऊपर वाला part is going to form anterior daughter cell, and नीचे वाला part is going to form your posterior daughter cell, right? Now the anterior parts are going to get some things from the parent cell itself, and some it has to develop. Something they are going to get from the parent over here. They are going to get parent from the parent already present in the parent, and some they are going to develop. For example, here you can see new oral groove is formed. So it is developed by the daughter cell, not the daughter cell. While division, it is developed by the parent cell, right? Now, let's see. Okay, I forgot to tell you that anterior daughter cells are called as proter and posterior daughter cells are called opist. Now here let's see, under favorable conditions cell grows maximum and stops feeding. Until here we have seen same in the where we have seen longitudinal binary fission, right? Then first karyokinesis occurs, so obviously both the nucleus are there, so in both the nucleus karyokinesis will occur. When we talk about the macronucleus, it will the karyokinesis occurs second. First karyokinesis will occur in macronucleus. Micronucleus is going to be reproductively important. It is going to be reproductively important for the paramecium. So first micronucleus is going to divide, which leads into two genetically similar daughter nucleus, because all the cell cycle process is being followed for the division of micronucleus, right? It occurs through mitosis. But when we talk about the macronucleus, it occurs through amitosis. None of the cell cycle division is followed. Randomly there will be the cleavage and division of the nuclei, which results into two daughter nuclei which is not genetically similar. Now, the oral grooves start disappearing when when there is a division of nucleus. The oral groove will start disappearing. Here you can see in the picture, it starts disappearing. After that, new oral groove is formed for the division of this cell, right? Now, then cytokinesis starts. Cytokinesis, how it will occur? Transverse constriction, which appears in the middle of the body. It deepens and forms two daughter cells. One is anterior, other one is posterior. Anterior one we call it as proter, posterior one we call it as opus. Anterior one, proter, is going to receive anterior contractile vacuole, cytopharynx and cytostome from its parent itself. It is not going to develop anything, right? I mean, it is not going to develop these three things. It will get it from the parents directly. What it was developing the posterior contractile vacuole and the new oral groove. For example, see here in the picture. So it is going to get three of the particles from the parent, then oral groove it is developing, and posterior one it has to develop, right? Posterior contractile vacuole it has to develop. Now when we talk about opus, it receives only posterior contractile vacuole, so rest of the things it has to develop, that is anterior contractile vacuole, cytopharynx, cytostome, all these things, and new oral groove, all these things it has to develop, right? Now, binary fission is completed in almost 2 hours. In favorable condition, Paramecium can produce 4 generation of daughter cell in a day. What is the meaning of 4 generations? So, this is your 1 Paramecium. After 1 generation, it will produce 2 daughter cell. After 2 generation, it will produce 4 daughter cell. And after 3rd generation, it will produce 8 daughter cell. And after, let me write it, and it will produce 16 daughter cell after 4th generation, right? Now, when we talk about the difference between proter and opus, it is nothing but it is the anterior daughter cell and this one is the posterior daughter cell. Proter receives what it is going to receive: anterior contractile vacuole, cytopharynx and cytostome from the parents itself. It is not going to develop it. The proter develop posterior contractile vacuole and new oral groove, right? And when we talk about opus, it receives only and only posterior contractile vacuole. Rest all the other things are going to be developed by the daughter cell, right? Now that's all we have in our transverse binary fission. Next, multiple fission we will see in next class.